Hello students, you are welcome to another class on EHS316, Immunology and Immunization. Now, this class promises to be an exciting one where further issues on immunology and immunization will be discussed. My name is Frank Iramoseli Ewudari, and I am your facilitator for this course. You are welcome once again. So once again, this is EHS316, Immunology and Immunization. That's my name, Dr. Frank Ewudari, and that is the email address that you can reach me in case you have any inquiries to make whatsoever. So for today's class, we shall be discussing the ABO blood group system. The ABO blood group system. And at the end of the class today, the learner is supposed to know how to classify blood groups. The learner should know how to classify blood groups. So, as we start, what exactly do we mean by blood? We must have been hearing that word, blood. Blood. What exactly is this blood that people talk about? What exactly does it mean? Let us go ahead. Blood is defined as the body fluid in humans and other animals that delivers the necessary substances such as nutrients and oxygen to the cells and transports metabolic waste products away from those same cells. Blood, as you can see, is a medium. It is the body fluid, a type of body fluid that exists in humans and other types of animals that help to deliver nutrients and oxygen to the cells, and also helps to remove or transport away metabolic waste products, that is, those products that are formed during cell metabolism. A normal blood supply helps to take away those waste products too so that the cells can function properly. That essentially is the meaning of blood. So we go ahead to discuss the different blood group and blood type. Or before as we go ahead, what exactly do we mean? Is there any difference between blood group and blood type? Can we say blood group is the same thing as blood type, or there is a difference between blood group and blood type? So what do you think? There is actually no difference between blood group and blood type. This simply means that both of them can be used interchangeably. You can use either blood group in a sentence or blood type in a sentence, but essentially both of them mean the same thing. So when you see blood group, you can still say blood type. Or when you see blood type, it means the same thing as blood group. This takes us to the next question. Do all humans have same blood group? There are over 7 billion humans in the world, and this question is asking if all these 7 billion people have the same blood group. Well, the answer is no. Not all humans. All humans do not have the same blood group. They have different blood groups. And that will take us to the next question. What are the different types of blood group system that we have? Types of blood group system. Essentially, there are two common types of blood group system. The first one is the ABO blood group system, and then the second one is the resource, that is the RH blood group system. So there are two common blood group systems, the ABO system and then the RH system. In the next slide, there's going to be a class activity. So please take a few moments of your time to attempt this class activity. So we continue. Now let us discuss the ABO blood group system. The ABO blood group system is the type of system that has about four blood group type. That is blood group A, blood group B, blood group AB, and blood group O. I'll take that again. Blood group A, blood group B, blood group AB, and blood group O. The reason why they are different is because of the presence of antigens on the surface of each red blood cell. That simply means there are some antigens that are like blood group A that make them to be classified as blood group A. Then there are some antigens that have different properties from blood group A then we now classify them as blood group B. And then there's still some type of red blood cells that have the A and B antigens on the surface, and that is blood group AB. Then of course the blood group O does not have any antigen on its surface. So this shows exactly what I just talked about, explaining the reasons why we have four different types of blood group system, which is based on both the antigen and antibodies that are present in the blood of any particular human. So that simply means any human being will have one out of these four group system, one out of these four blood group system, that is blood group A, blood group B, blood group AB, or blood group O. So for an individual that has blood group A antigen on the surface, the corresponding antibodies that will be found in that person's blood will be anti-B antibodies. That simply means an individual cannot have both antigen and antibody existing together in the same body. For example, an individual cannot have blood group A and antibody A existing together in the same body. This will not support life, because there will be a transmission reaction that can lead to the death of that human being. So what we have is this: if an individual has blood group A antigen on the surface of the red blood cell, then the antibody that will be present in that individual will be anti-B antibodies. That simply means that if any blood, for instance, any blood group B is transfused into that human body, antibodies that are present, anti-B antibodies present, will react to that blood group B and then cause transfusion reactions. So in essence, blood group A, we have anti-B antibodies. Blood group B, we have anti-A antibodies. Then for blood group AB that have both that has both A and B antigens, there are no antibodies for blood group AB. While for blood group O, there are both anti-A and anti-B antibodies. Now, we shall discuss the second classification or the second type of blood group system, which is the RH system. So an individual can either be RH positive or RH negative. They are both mutually exclusive. That simply means an individual cannot have both RH positive and RH negative system. It is either you're RH positive or RH negative. So, because of the ABO and RH system, there is usually a combination of these systems for any individual. That is, any individual at any time will have a combination of both the ABO system and the rhesus system. That makes a possibility of 8 types of blood group system after combination of the two. You can either be A positive or A negative, B rhesus positive or B rhesus negative, AB positive or AB negative, and O positive or O negative. So I repeat, an individual can be one of these 8 combination, cannot be any two of these combination. So an individual can either be A positive, or it can be B positive, or it can be AB positive, or even O negative or O positive, or it can be A negative, and so on. So that is how the ABO and rhesus system can be combined to give us the blood group that we actually are known to have. Now that we have identified, or we have established the different blood groups in the body, how then do we identify that an individual has a particular type of blood group or a particular blood group in his body? How is this essentially done? How can I say that? How can I identify that a patient or an individual is A positive or A negative, or is B positive or B negative, and so on? How is this done? The way this is done is agglutination reaction. Agglutination reaction. That is when a particular surface antigen reacts with its antibody to cause a sort of reaction. That helps us to identify which particular blood group that is available in that individual. When antigens react with its corresponding antibody. So for instance, if an individual who is blood group A reacts with the blood containing antibody A, that helps us to know that that individual is blood group A. I'll take that again. If the blood group, if we bring, you take a blood sample of an individual and then match or react them with known antibodies, if there is a reaction of some sort that leads to precipitation, that simply means if, for instance, the antigen that is in the blood, for instance, is A, and then when this antigen reacts with the antibodies on the plate outside of the body, any reaction that occurs with the corresponding antibody will help us to determine the blood group of that particular individual. I'll take it again with another example. For instance, if a blood sample is taken from an individual and that particular blood sample is made to react with known antibodies on a plate, it is usually done outside of the human body. So any of the reaction Any of the uh, combination that causes a precipitation, that is, that makes some solid components to precipitate to come out of the reaction, that is how we can now tell what exactly or the type of blood group that is in that particular individual. Any reaction that leads to precipitation helps us to identify the blood group of the individual. So in the next slide, there's still yet another activity that you should carry out. So please take a few minutes of your time to carry out this activity. This leads us to a concept called blood reactions during blood transfusion. That is, reaction that occur when a patient is being transfused with blood. If blood containing a type of antigen, for instance blood group A, is transfused into another blood and into a person that has blood group B, that is, this person has A antibodies in the system, there will be a reaction. If an individual has a blood group, a certain blood group, for instance blood group A, and this blood is transfused into another person that has another type of blood group, let's say blood group B, that already has A antibodies in it, these antibodies will react with the antigens in the blood group A to cause blood transfusion reaction. So it simply means that for us to now have safe blood transfusion, we should have a serum of the patient that does not contain antibodies for the blood cell antigens of the donor. I'll take that again. For us to not have blood transfusion reactions, this requires that the serum of the patient that is receiving the blood should not contain antibodies for the blood cell antigens of the donor. So, in conclusion, we have been able to discuss the different classification of the blood group systems, and then we've also known that this difference is as a result of the difference of the antigens on the surface of the red blood cells. Until next class, I remain your facilitator, Frank Eromaseli Ewudari. It's goodbye for now. Thank you.